So I'd like to start by thanking the organizers for this invitation to speak at the plenary. I'm delighted and very honored to be here. And I want to start with this disclaimer. I came very late to FASD research. And I want to lead you a little bit into what got me into FASD, including FASD in my research program. And I want to start by acknowledging the members in my department who have been responsible for this education for me, specifically Rajesh Miranda and especially Shamina Baik, who has spearheaded the studies that I will talk about today. So I study stroke, and I want to tell you what stroke is about. I'm sure many of you know about it, so I will keep this brief. Stroke occurs when blood supply to the brain is interrupted. And this can happen in a couple of different ways. You can either have an ischemic stroke where a clot occurs in a blood vessel, and the clot rolls around until it finally gets to a blood vessel whose diameter is smaller than the size of the clot, and it gets stuck there. And at that point, blood supply to the brain especially will be interrupted for everything beyond that clot that particular artery serves. This is the most common type of stroke. 87% of all strokes are ischemic strokes. The other type of stroke is a hemorrhagic stroke, and this is when the blood vessel has a weak wall and therefore blood leaks out into the brain. That has some very critical implications. Those are fewer strokes, and that is not the subject of the talk today. So I'm going to talk about ischemic stroke. What happens when you have an ischemic stroke? Essentially, brain cells are starved of oxygen and glucose. And as a result of that, they will die very, very quickly. The brain is very, very dependent on oxygen and glucose that comes to it via the blood supply. During a stroke, 2 million brain cells can die every minute. And this can increase the risk for permanent brain damage as well as disability. And no two stroke patients will look identical. For example, if you think of the other major diseases of our time, you have a pretty good idea of what an Alzheimer's patient might present with, what a Parkinson's patient might present with. But in a stroke, the type of disability will depend on which blood vessel was affected. So you could end up with a disability in your sensory system, in language and speech, or in motor function, depending on the blood vessel that's affected. Now, what I want to emphasize as we go along is that there are some long-term features of stroke that are independent of which blood vessel is affected, and that is what my lab is very interested in. What we've been studying for a very long time is sex differences in stroke. Stroke is the 5th leading cause of death in men, but it is the 3rd leading cause of death in women. Many more women will die of an ischemic stroke, and they will also have greater disability and more strokes as they grow older. I mentioned the long-term impact of stroke. We know that you can have health issues related to weakness or balance problems after a stroke, changes in your sensory systems. The issues that tend to come up much later are post-stroke seizures, post-stroke depression, and post-stroke cognitive disorders. This is the issue I want to focus on today because they tend to be independent of where the stroke actually occurs. So here is just a recap of the issues related to ischemic stroke. It is the #1 cause of disability outside of war. In terms of long-term cases, individuals with stroke will end up with post-stroke depression. More than half of them will have cognitive impairment, and it is one of the leading causes of dementia. This is the part that I think, as researchers, we have the ability to intervene and improve the quality of life. Now, I mentioned to you that I have been looking at sex differences in stroke. The biggest risk factor for stroke is age, and in the stroke literature is referred to as non-modifiable, unless somebody has found the elixir and the answer for long life. Sex is a major contributing element. Young women will have fewer strokes and they will do much better after their strokes. After menopause, women will have more strokes and more severe strokes. That is another clue about how stroke risk can be affected and perhaps be a point of therapeutic intervention. We also know that stroke risk is increased by type 2 diabetes, by hypertension, by high cholesterol, and several other factors, and I put asterisks there because this is what led me into looking at FASD and stroke. I'm going to put this slide up here, and nothing in this slide is new to anybody in this group. The only point I want to make about prenatal alcohol exposure is that there has been a lot of work and literature on the neurodevelopmental disorders and changes that occur as a result of FASD. And what I'd like to focus on today is we don't know very much about the chronic diseases that occur in adulthood and with middle age and with aging in this population, in part because we don't have a large enough cohort of people at all of these ages. I was very influenced by this book called The Lay of the Land, which has been referred to very often in this meeting. And one of the things that really struck me is the number of chronic conditions that were reported in this community-based health survey. And they include coronary heart disease, they include obesity, cerebral hypoperfusion, that is lowered blood supply going to the brain, metabolic disease, of which type 2 diabetes is a big part, complications related to the GI system, which Claire alluded to, things that have to do with just gut motility. And I particularly want to draw attention to this because of the new data on the gut-brain axis. There is a bidirectional communication between the gut and the brain. And what happens in the brain influences the gut and vice versa. And I mention that because that is a target that we can use to intervene to improve the health of people. Premature menopause, because of my interest in sex differences and the fact that menopause is a risk factor for stroke, I was very struck by the information in the Lay of the Land about premature menopause among women. All of these sort of convey to me that these might increase risk factors for stroke among this population. And so I'm going to show you a modification of the previous slide, and that is another factor that could influence how people recover from stroke or their risk for stroke is FASD. So we know very little about the risk for stroke, and especially stroke-induced cognitive impairment and dementia in FASD. And that data is starting to emerge from a number of sources. And again, in the Lay of the Land, there was an estimate that early onset dementia, which is about 0.008% in the general population, rises to 0.9%. This is a 200-fold increase, and this is data that needs to be looked at very carefully. So all of the work I'm going to show you today is in a preclinical model. It's in an animal model. And I'd like to take a second to explain the utility of an experimental model in PAE and stroke. Over yesterday's pre-con session, as well as today, we've heard about the number of coexisting conditions that happen with FASD, the ACEs, and ACEs-A that occur with adulthood. What the preclinical models allow us to do is to understand what is the specific effect of FASD, because we can hold everything else constant. We can hold the living conditions constant, their food, our animals get excellent health care. We can control all of those conditions. What varies, obviously, is the exposure to FASD. But we also know that FASD itself, even in our preclinical models, will result in whole body changes, such as changes in glucose tolerance, changes in hypertension. And that is another clue that perhaps those might be related to the fetal alcohol exposure that increases the severity of stroke. If you were able to attend the sessions earlier in the day, Shamina Baik presented data on how prenatal exposure itself results in behavioral changes, and she tied that to changes that occur in inflammatory cytokines. Nadia Samia, in that same session, described data of prenatal alcohol exposure in a transgenic rat model and showed that it does influence certain or accelerate certain cognitive impairment. And what I'm going to do right now is tell you about our work in the stroke model, which is essentially the middle cerebral artery occlusion model. Okay, and within that model, I'll focus on a test of post-stroke depression and a couple of tests on post-stroke cognitive impairment. Much of this work is published. Some of it will be out pretty soon. And so I'm just going to focus on a couple of highlights to tell you what we've learned. Now, how do you create stroke in an animal? You can't wait for an animal to have a stroke. Nobody would ever graduate. So we create a stroke by going in there and actually interrupting blood flow in a vessel. And in the model that we use, it's called an MCAO. If you read this literature, what that simply means is middle cerebral artery occlusion. This is one of the major sources of blood supply to the forebrain, both in rats, mice, and in humans. And it provides blood supply to the striatum and the overlying cortex. And what you're looking at over here is injections of a vasoconstrictor that constricts this vessel, and on the right side you will see 3 slabs of brain sections. The unstained portion is the part that died as a result of the stroke. Okay, that's called an infarct. And I'll be talking about infarct volume. That tells you about the extent of brain damage that occurred. And I want to be sure that I mention this caveat. In our preclinical studies, we cannot estimate risk. What we are looking at is severity. So because we caused the stroke ourselves, what we can assess is how bad is the stroke? How much is the animal impaired, whether it was exposed to FASD or not? Now, this is a very confusing slide, and I'll walk you through it pretty quickly. This study basically encapsulates what we did. Animals were exposed, that is dams, pregnant dams were exposed to alcohol in a vapor chamber. We chose that model, even though it has some pros and some cons, but having to do an oral gavage on a pregnant dam every day for so many days, we were worried would be a problem. And then the offspring were tested at 6 months and at 12 months. So we did a stroke at 6 months and we did strokes at 12 months, and we followed them, these animals, for about 90 days. And here is the data that we have. I put this in here because it illustrates a point I made earlier about data in the lay of the land. In our PAE exposed animals, the females have lower levels of estrogen. Estrogen is very neuroprotective for stroke in younger animals, so this bears watching. We also looked at IGF, another neuroprotectant that my laboratory studies, and that is also affected, but mostly in males. So what did we see? What we saw, whoops, I need to go back. How do I go back? This one? Yes. When we create the stroke at 6 months in adult animals, we actually did not see any effect of PAE. Males had larger strokes, females had smaller strokes. We've always known this. No effect of PAE. However, if you do tests of motor, that is how impaired were they on motor function, you start to see that the PAE did impact the motor function. If you do this stroke in middle-aged animals, and this is an important group because in most of human stroke data, the risk starts in middle age, but we wanted to be sure to look earlier as well in case FASD accelerates the stroke risk. And here we find that in middle age, animals that had a prenatal exposure had much larger infarct volumes as compared to those that were not exposed to it. We looked at depressive behaviors. This is a social interaction test. Rats are very sociable animals. They enjoy interacting with another rat. A depressed rat will not do that. And what we found is that after a stroke, 90 days later, males decrease their interaction with another rat. Females actually don't. PAE accelerates that loss of sociability in males. This is in the middle-aged animals. We didn't see any effect at all. Okay? The data is very variable, and it just shows that aging does modify the impact of stroke. We looked at cognitive performance in a task that requires you to have an idea of the spatial orientation. This is the Barnes maze. And it allows you to test both learning as well as recall. And what we found is that in males, there was an impairment in learning. They were unable to find the escape hole in the Barnes maze, and the females were actually impacted by recall when tested a day later. I want to stress this point. We are seeing a lot of impairment that occurs in one sex but not the other, and at one age of stroke but not the other. Similarly, in another memory test, we found that, in the interest of time, I will skip this. This is already published data, and move to my conclusions. What we find is that PAE impairs specific aspects of stroke-related outcomes and cognitive affective behaviors. They tend to differ in males and females, and these outcomes may be more severe if you were to add other ACEs or ACEs-A factors, which we did not do in this study. And finally, many of the comorbid conditions for stroke that are elevated in FASD can be handled by medical management for hypertension, for example, type 2 diabetes, etc. And I mention this because it offers some hope that one could ameliorate or alleviate the risk for stroke outcomes by these strategies that are already well established. So I want to thank you for your attention, and I'll be happy to take any questions.
We do have time for one or two questions.
Yes.
Thanks. Just a quick question, because we've heard a couple of times about premature aging. And I was just wondering, do we know how prenatal alcohol works on telomere length, and whether that has an increased vulnerability to cells, and whether that is leading to increasing the likelihood of having premature death to those cells?
That's a really great question. I know there are attempts to look at telomere length in the context of not only FASD, but other neurodevelopmental teratogenic exposures. One of the things that I believe is contributing to the premature aging is the impact it has on immune cells. As Charlie's mentioned, an overactivated immune system can drive some of these behaviors. In normal aging, okay, there is a phenomenon called inflammaging. Inflammation increases with age irrespective of other conditions, and that may be accelerated by FASD. So there are a number of possible targets, but telomere length would be a really good one to study.
Okay. Great.
Thank you.
Thank you.